Hi, my name is Helene Newsome. I'm a BSN student working under Dr. Lawman and working on my Master's in Science and Nursing degree. And I will be presenting to you a lesson on liver and cirrhosis today. So I will go ahead. That is part of your Threats to Nutrition module, and that starts, I think, in your book on page 986. Briefly, we'll review the anatomy of the liver, just to refresh our memories on where the liver is located in relation to other organs. The liver is located in the upper right-hand portion of the abdominal cavity, beneath the diaphragm and on the top of the stomach, right kidney, and intestines. Shaped like a cone, the liver is a dark reddish-brown organ that weighs 3 pounds. The liver weighs approximately 3 pounds. There are two major lobes, a right lobe, which is the larger one, and the left lobe. Each lobe is then divided into several smaller lobules. The liver is supplied by two major blood vessels on its right lobe, the hepatic artery and the portal vein. The hepatic artery delivers oxygenated blood to the liver. The hepatic portal vein carries deoxygenated blood rich with newly absorbed nutrients from the small intestine, spleen, and stomach to the liver. This vessel is anatomically located in the inferior aspect of the liver. So just to show you here in this diagram, you've got right here your right and left hepatic arteries, and then right here is your portal vein. So that is the two major blood supplies. And then both oxygenated and deoxygenated blood enter the liver into the common capillary bed called sinusoids, located between the plates and the hepatocytes. Okay, so you're going to be entering into the liver. The blood flows from the sinusoids to the central vein to the hepatic vein. So here's your hepatic vein up here. And finally into the inferior vena cava. So that's a general rundown of how the blood flows. The hepatic duct delivers bile to the common bile duct from the liver. The cystic duct delivers bile to the common bile duct from the gallbladder, and the common hepatic bile duct passes into the duodenum to deliver the bile. So in this picture, you can see a very general view where you have your hepatic artery, your hepatic vein, and then you're seeing where your gallbladder is, which is, let me go ahead and annotate here. So your gallbladder again right there, and then you've got your common bile duct. Okay, so the bile is a fluid that is made and released by the liver and stored in the gallbladder that helps with digestion. It breaks down fats into fatty acids, which can then be absorbed into the bloodstream through the digestive tract. Bile contains mostly cholesterol, bile acids, and bilirubin, which is a breakdown of product of red blood cells. It also contains water, body salts, copper, and other metals. So that is how, what bile is made up of. Now the liver is made up of hepatocyte cells, which is a cell tissue in the liver. It's in the cell tissue in the liver, and it makes up about 55 to 65% of the liver's mass. These cells are involved in protein synthesis, protein storage, and hepatocytes also produce and secrete bile. They are able to regenerate themselves, therefore damaged or resected liver tissue can regrow. As we've heard with cirrhosis and other liver damage, there is some regrowth that occurs for a period of time, and we'll go through that more during this lesson. Now in this diagram, you see here, it gets a little more where you can see the hepatocyte on the right-hand side of the diagram, and I can annotate that a bit. So you've got your hepatocyte all in here. These are your hepatocytes. And then the Kupffer cells, also known as macrophages, are specialized, located in the liver, the lining walls of the sinusoids. So here's your sinusoids, okay? They form part of the mononuclear phagocyte system. So they act as, basically they engulf the foreign bacteria. These cells are also found in the spleen, lungs, and lymph nodes, and they are needed for conjugation of bilirubin, the byproduct of destruction of aged-old red blood cells. Now, the liver's main function is to metabolize carbohydrates, lipids, and proteins to maintain a normal glucose level in the blood. When the blood glucose level is low, the liver breaks down glycogen to glucose and releases it into the bloodstream. The liver can also convert certain amino acids and lactic acid to glucose. When the glucose level is high, especially after ingesting food, the liver converts glucose into glycogen and triglycerides for storage. Now this picture shows the breakdown of protein. So basically, in the metabolism, glyconeogenesis is the process of forming new glucose from non-carbohydrate molecules within the liver. Now, glycogenolysis is the breakdown of glycogen stored in the muscle and liver to release glucose into the bloodstream. And then glycogenesis is conversion of glucose to glycogen for storage within muscle cells and the liver. The byproduct of protein metabolism is nitrogenous waste. So nitrogen will very easily bind with hydrogen forming ammonia, which is very toxic to our cells. Ammonia is then converted into urea by the liver, and it passes into the blood, where it is then excreted from the body by the kidneys. So in this diagram, as you can see here, you've got your ammonia, and then urea goes into the bloodstream and gets excreted through the kidneys. Now, this is the normal process. However, if there is an abnormal amount of protein synthesis, the ammonia will build up and can damage the liver and cross into the blood. It will do that. It will also cross the blood-brain barrier, causing irritation to the central nervous system. And we've heard about that with cirrhosis, that can be a problem. The buildup of neurotoxic substances within the blood leads to hepatic encephalopathy, which we'll talk about more as we get into the complications with cirrhosis. So the liver is responsible for synthesizing and producing proteins, such as albumin as well, which has a lifespan of about 14 days. The liver also synthesizes our clotting factors, prothrombin and fibrogen, and the aid of vitamin K. The clotting factor's lifespan is 24 to 36 hours. The liver also produces lipoproteins, which are essential in order to maintain cell membrane integrity. Albumins are proteins that maintain the isotonic environment of blood, where cells of the body don't gain or lose water in the presence of body fluids. Also, the liver stores many useful nutrients, vitamins, and minerals obtained from the blood passing through the hepatic portal system. So immunity is another function of the liver as well, and it is an organ of the immune system through the function of the Kupffer cells that line the sinusoids, as we discussed. Kupffer cells are the type of fixed macrophage, so we already discussed that as well. Now, the production of bile is a substance used in the emulsification of large fat molecules into smaller particles to aid in the digestion of lipids. Now, conjugation of bilirubin in the spleen, old red blood cells are broken down into three parts. Globin, which is the protein, ferritin, which is the iron, and heme, which is the pigment. The globin and the ferritin are recycled, but the heme is transported to the liver as unconjugated or indirect bilirubin, which is not water soluble, but lipid soluble. Within the liver, indirect bilirubin is conjugated with glucuronic acid to form conjugated bilirubin, or direct bilirubin, which is water soluble, by the hepatocytes. Okay? This allows for the bilirubin to move into a water solution. The conjugated or direct bilirubin is then secreted into the bile and moves to the small intestine through the biliary ducts. In the large intestine, bilirubin is converted to urobilinogen through bacterial action. And this is what eventually gives our feces a brown color. Patients who have biliary blockage or liver disorders may have a slightly lighter color to their feces, or a foul smelling as well, kind of fatty, foul smelling. The liver also is involved in the activation of vitamin D, which is essential for the absorption of calcium from the intestines into the bloodstream. The liver makes vitamin D convert to calcidoid, which is then converted into the physiologically active form within the kidneys. This process also requires exposure of skin to sunlight.